All right, fellas, welcome back. I'm sure you're missing your favorite class of the year, so I figure we get some notes going. For the next two chapters, we're going to see, or for this one and the next one, we're going to see how this is going. If you guys don't like it, we can change up a little bit. We are going to have a test. We're going to have a Unit 4 test on these next two chapters. It will probably be a Schoology online test. We'll see how that goes, and then we'll go from there. If we need to adjust, we can talk about that as well. I thought you guys would have wanted a Zoom meeting. We could all see each other. I know you guys must miss uh, the dojo as well, but we'll get started today on our DNA fingerprinting. Our DNA fingerprinting, basically what we're going to go over, how we're going to do it. It's not just fingerprints either. It's basically just getting DNA from different types of trace evidence that we can find, blood, semen, anything like that as well. So as we go through this chapter, we're going to see a bunch of different things. You guys already did a few assignments. You did the vocab, you did the chapter review, so this should be very familiar to all of you. So hopefully we can get this started and should be going over quickly. All right, so history of our biological evidence in forensics. It's called our DNA fingerprinting, but it's also known as our DNA profiling. It's going through the different types of DNA that we can have. And this is going to be extremely helpful for us in forensic science because it does have a very, very, very high degree of accuracy. The percentages you can see when you test DNA, it's very difficult to disprove. And this is because it's going to be specific to different people. Like we said, fingerprints are unique, but your DNA is also going to be extremely unique to you as well. Some forensic laboratory techniques were originally developed for other purposes. Can you name any of them? And also, I'm probably going to be going through this quickly. If you need to, you can pause the video as well to get those notes filled in. How this DNA is going to work, I'm sure you guys all remember from lovely bio class your freshman year. Our DNA strands that we have contain genetic material of our cell. Chromosomes are located in the cell nucleus, and those chromosomes contain long DNA strands wrapped around proteins. And you can see that double helix that we have there above. And it's probably bringing back some good memories with Miss Bliss if you had her, Mr. Bruno, or Mr. Semenek. Some of your chemistry grandfather. How our DNA is going to look, it's our nitrogen bases. All of them are going to be pairs of molecules that form the rungs of the DNA. Or sometimes say our ladder. They're the four types, and if we look back to the previous slide, you can see in the double helix you have your C, your G, your T, and your A. Notice that anytime we have a blue tab, that's going to be our A. It's going to be matched up with a T. Our T's are always matched up with our A. And then our green tabs with our G are going to always be matched up with our C. And what those A, T, C, and G mean, our A is for our adenine, which is always going to be then matched up with our T or our thymine. And then our C cell, which is going to be our cytosine, is matched up with our guanine. Jump the gun a little bit, and you can see here, our adenine is always going to be bonded with our thymine, and the same will be for our cytosine and our guanine as well. How we're going to be able to identify some of this, or some of our DNA, our polymorphism, it's going to be non-coded DNA. Basically, what we just saw, it's not going to have that A, T, C, and G that contain unique patterns of repeated base sequences that are unique to individuals. Our poly, prefix poly, means many, so there's going to be millions and millions and millions and millions and millions of sequences that you can go through to see that. Our DNA profiling is going to be able to isolate and then analyze those polymorphisms. Like I said, there's going to be those millions and millions and millions different, like almost like code on a computer. We're going to be able to track to match up certain unique pairings that you can see from two different samples. The DNA fingerprints appear as a pattern of bands on X-ray film. We'll talk a little bit more about that as well. There are two types of our DNA sequencing. Two types that we're going to look at is going to be our variable numbers of tandem repeats, our VNTR, and then our STR, which is our short tandem repeats. The short tandem repeats exactly what the title says. It's short tandem, so it's only going to be about 2 to 5 bases in length. This is going to make it a little bit easier to use than our variable number of tandem repeats, just because that's going to be a much larger sample that we have as well. For our variable number of tandem repeats, the number of repeats varies from person to person. Like I said, because we each could have this unique set of DNA. Both of them, though, are analyzed for tissue matching and inheritance matching. You're supposed to get 50% of your genes from your mom, 50% from your dad, so you're going to be able to match up those different strands between your two parents. Once we have the tissue matching, while we're going to be able to use that, we can take one sample and we can test it with the different persons as well. Two samples that have the same band pattern are from the same person. So if we have some type of evidence from the crime scene and we're able to match it up with someone else's DNA, maybe a suspect, then we can then fairly certainly assume that that person was at that crime scene. It might not mean that they did whatever the crime was, but they were at the location. And then our inheritance matching, each band in a child's DNA. That's supposed to be DNA, not NDA. Fingerprint must be present in at least one parent. So again, you get 50% of your genes from your mom, 50% from your dad. So at some point, you're going to have matching pairs from them. Doesn't mean that all of your DNA is going to match with them, but 50% will as well. Population genetics, the study of gene variations among groups of individuals based on where they live. Difference between someone here in the United States or someone here in Pennsylvania and maybe someone in Alaska, or someone from Europe, or someone from China, or someone from Africa. They all have those different characteristics that we have. We look different. We have different hair types. That's going to be based off those population genetics. This analyzes the probability of a random person with the same alternative form of a gene, which we call an allele. Examples of why we would need some of this DNA sequencing to identify a suspect in a crime, or identifying an alleged father in a paternity case. I'm sure everyone watches Jerry Springer. That's how they do their paternity tests as well. Sources of DNA that we can use, examples include saliva, blood, skin, hair, semen, any of those that you can get DNA from. Is going to be some of our biological evidence that we have. It's going to be unique to each individual, and each individual evidence is capable of identifying a specific person. It could be a very, very, very small amount that you can test, but it makes it easier the more sample you have. And we did hair a little bit earlier this year. Again, we know where all of the DNA is, or we talked about where most of the DNA is in our hair sample. Then, again, our small amount may be only trace evidence, difference between trace evidence, class evidence. And then our polymerase chain reaction technique generates multiple copies of DNA evidence. Our PCR, we were going to do our PCR this year, but I guess we will not be able to now. How we get collect and preserve. This is pretty self-explanatory and shouldn't be anything new for most of us. Make sure you're wearing gloves when you collect. Again, we don't want Locard's principle of exchange where we're changing our evidence or our DNA onto their DNA. Do you want to avoid physical contact, talking, sneezing, coughing? Hey, look, just like coronavirus. Then you want to make sure that your evidence is air dried, put it into a paper bag or an envelope, depending on what you have. If it's a wet sample, make sure that it goes into a test tube as well, or a vial. Then, depending on when you're going to be able to test it, you want to make sure that you dry out the evidence or freeze the evidence. If it's something like blood or semen, you don't want to dry it out. And then keep the evidence cool and dry during transportation and storage. And prepping our samples for fingerprinting, DNA is mixed with special enzymes. We kind of saw this with our online electrophoresis lab. You're going to prep the DNA with that special enzyme, and the enzyme's going to be able to break it down so that way it's going to be able to travel through that gel that we had. Enzymes cut apart the DNA in specific places, forming different sized fragments. Since all of you did your online lab for the gel electrophoresis, you saw those different, almost like spots that they had lined up, and that's what our enzyme's going to do. The DNA is separated within the agarose gel, which is just that clear film that we had as well. Then electric currents pass through the gel, separating the fragments by size, and we're going to talk about our gel electrophoresis in a little bit too. Extraction. Cells are going to be isolated from biological evidence such as blood, saliva, urine, semen, and hair that we talked about already. They are disrupted to release the DNA from proteins and other cell components. And then the DNA can be extracted from the cell nucleus, so that way we're able to see the different patterns that are going to be created from them. How we're going to be able to see them, it's actually based off the mass of those samples. Whichever ones can travel the furthest are going to be the lightest as well. And again, prepping our sample for our VNTR analysis, polymerase chain reaction can be used to amplify the DNA that contains the VNTRs, again the variable number, and then our short tandem is going to then be able to then uh, allow our amplification with strands of the STR sequences. And like I said, our electrophoresis. Electrophoresis is probably going to be the most common. We also had our lab set up. Some of you guys might have done this with Mrs. Bruno as well. Basically, our electrophoresis, you have that gel. I don't know if you can see my mouse, but if you look at the picture in the top right corner, you see that slab of gel. Then you load in your sample into different slots that they have on there with a micropipette. Electrophoresis is the DNA samples are being placed into gels through which electric currents are passed. Then the DNA fragments line up in bands along the length of each gel. You can see almost like a spotted color pattern to it as well. Electric apparatus running 5 samples of DNA. You can see, starting off with our loading point, that eventually it's going to all travel throughout, and you can match up the different patterns. If, say, sample 1 was our suspect, and you had 4 different DNA samples they wanted to then analyze, you can match it up, see which ones are going to be able then to have both DNA samples or those fragments in the same area. And then arrows show the movement of the negatively charged DNA fragments through the gel matrix. Our DNA probes, like I said, you can see those different spots that you can match up with different people. You can see that there's a spot here, a spot there, and then match up as well from there. Our DNA probes, they identify the unique sequence in a person's DNA. They're made up of different synthetic sequences of DNA base complementary to the DNA strand, and they're going to bind to complementary bases in the strand. You can see that above here. You can see some of them don't match up at all, or there's only 1 or 2. You want to make sure that you have at least or around 50% of those DNA samples matching up with them again, because we said the sample 50% from mother, 50% from father. If you're looking at a crime scene, say if this was some DNA collected at a crime scene, and these were 3 different suspects. Well, if this is somebody's, then it has to match up exactly, almost, with their DNA strands. Now, this could also be just for a paternity test as well, to match up 50% of the DNA strands with someone else's. And like we talked about, you can see here we have a 50% mother, 50% father, matched up the first row that we have. Then we have our third row, and then they both have our last one as well, which is going to be common too. And how we're going to be able to use our DNA, how we're going to be able to apply it to our forensic science. Basically, some pretty simple ones. You can match crime scene DNA with the suspect. If you collect some blood that is left at the scene of the crime, and you have 5 different suspects, you can then take those samples, compare them, and then see which ones match up best. Determine maternity, paternity, or match to another relative. Again, think Jerry Springer. That's how they're going to be able to do their paternity test as well. You can also have the flip effect as our first one. You can eliminate suspects. If DNA doesn't match up, then clearly they weren't there in order to transfer that DNA. Or free a falsely imprisoned individual. I'll probably say this is a little bit more uncommon because you can voluntarily give a DNA sample. Again, if you're innocent, why would you not do it? If you're guilty, why would you do it? But maybe try to trick the system. And then you can also use this to identify human remains as well. And I believe that is the end of this chapter. I know that was kind of quick. If you want to slow that down, or I'm sure on YouTube you guys can speed it up to 1.5. I don't know if you're going to need to, but try it out. If you have any questions, please let me know. And if you guys would like a Google Hangout, we're more than welcome to. We could do a Zoom meeting. I'm on Discord. Please check into Discord. If you do not know what Discord is, that's probably because you haven't signed into it yet. Make sure you go back to our updates on Schoology. Check in there. I'll be on there from 9 to 12 today. Have a nice day, gentlemen. Stay safe.